Hello, my name is Thi Duyen and I'm with Taipei Medical University. My research primarily focuses on using Pan-genome as a genome representative method and employing machine learning to predict antimicrobial resistance (AMR) in Pseudomonas aeruginosa, while also investigating novel AMR mechanisms. AMR is the leading cause of death among various health threats, with an expected death toll reaching 10 million by 2050. Notably, Asia and Africa are projected to face the highest number of AMR-related deaths, with Asia estimated to account for 4,730,000 and Africa for 4,150,000 deaths annually. This staggering number highlight the urgent need for proactive measures to combat AMR. This research aims to develop an AMR predictor that can forecast AMR without relying on wet lab experiments and a single reference genome or even AMR databases. Additionally, we strive to reduce data complexity while enhancing the model explainability. The AMR mechanisms of Pseudomonas aeruginosa are diverse and complex. Pseudomonas aeruginosa can employ various strategies to protect itself, such as using efflux to expel toxins from the cells, employing quorum sensing, and two-component signal transduction system to detect and avoid toxic areas. Additionally, they can spread AMR genes through horizontal gene transfer to develop mutated strains. The workflow includes several steps. First, Pseudomonas aeruginosa whole genome sequencing data was downloaded from various sources for training and external validation, followed by a genome quality check to filter out contaminated and incomplete genome. Next, protein sequences were grouped into gene clusters to generate a gene cluster pangenome. Protein alignment was then conducted for each gene cluster to identify different amino acid positions. Subsequently, we mapped these positions back to DNA sequences to find SNP position and construct a SNP pangenome. We also created a combined pangenome that include both SNP and gene cluster information. We use edibles to preliminarily filter out noises and less important features. Next, we employ a support vector machine incorporated with a genetic algorithm to select the most important features. We hope to explore novel and significant AMR mechanism from these features. Genetic algorithms are heuristic search methods inspired by natural selection. They start with an initial population of potential solutions represented as binary factors. Each solution's fitness is evaluated based on its ability to predict AMR using SVM models. Selection favor solutions with the higher fitness, allowing them to reproduce and pass their genetic material to the next generation through crossover. Mutation introduces random changes, maintain diversity. The process iterates for 3,000 iterations, gradually refining the population toward better solutions by combining successful features and optimizing predictive accuracy. We found that the gene cluster pangenome is an open pangenome, indicating the remarkable diversity within this bacterium's genome. We found that the three pangenome approaches demonstrated comparable performance on the training datasets. However, during validation, the combined pangenome approach, which integrates gene cluster present absent patterns and SNP information, outperformed individual methods. It achieved an average AUC of 92.3% for the training datasets and 76.5% for an external validation dataset, exceeding the SNP Pan-genome by over 7.5% and the gene cluster by over 9.5%. Our proposed method and edibles both excel in identifying crucial features for AMR prediction, outperform the other two feature sets across all pan-genome approaches. In addition, the feature subsets identified by GASVM include about half of those selected by Adaboost, indicating that our model effectively identified the most crucial AMR determinants while maintaining comparable prediction accuracy to Adaboost. The GASVM and Adipose model both perform well in predicting AMR, with GASVM showing a notable improvement in the average F1 score and an MCC that is 3.3% higher than that of Adipose. This indicates that our model, which evaluates multiple metrics, including accuracy and F1 score, demonstrates enhanced robustness. Our results demonstrate that integrating gene cluster and SNP present-absent information is a more effective predictor of bacterial pathogen resistance than using either alone. Our two-step feature selection-based GA identify highly relevant features for AMR prediction, significantly improving the F1 score and reducing the number of features. This framework aims to explore underlying AMR mechanism and enrich the resistance repertoire by interpreting the contribution of each AMR indicator.